Well, this is a crucial part, and it's really is worth investing some time with your students so they know about how to use the various pipettes and the ones, particularly ones you're going to use with them during the whole process. So back over to Mary again, who's going to lead us through the different ways that each of the pipettes work and how vital it is to follow the correct procedures with them.
Okay, now the principle of using pipettes is the same as using one of these little plastic pipettes that I'm sure you're familiar with. The idea is that you push air out of the bulb and then you insert the pipette into your liquid and you draw up the volume that you need. But for DNA work, the volumes are very much smaller. So for the little plastic pipettes that come with the kits, the idea is that you would draw up just enough liquid following the instructions of the kit. And after you dispense that liquid, you would then throw that pipette away. For the reusable pipettes, the simplest fixed volume pipette, for the use of the fixed volume pipette, you push down the plunger to where it stops, put your pipette into the liquid and then release the plunger. And in the tip of the pipette you can now see a very small volume of liquid, and it is a complete bubble. There is no air in that liquid in the tip, and that is measuring 5 microliters. You would then dispense that liquid into your container, and afterwards you would remove the tip and discard it.
Okay, so that's vital, isn't it, that we don't contaminate tips with one thing or another.
It's really important for DNA work.
But the tips get discarded.
Yep.
Okay.
Now, for the fixed volume pipettes, I'm going to use the larger fixed volume, the variable volume pipettes. I'm going to use the larger variable volume pipette, the sort that a technician would be using to dispense materials for the class. Now, the variable volume pipette operates by, if you twiddle this knob here, the numbers on the side here give you the volume that you will be measuring at into the tip. And let's say that the technician has been asked to measure 20 microliters. You just twiddle the knob until you get 20 microliters. In the window. Now, what's really important is that the technician always ensures that the range of the pipette is not exceeded. The range is always written clearly on the pipette. You must not use the pipette beyond that range, or you will damage the pipette. Now, to draw up the liquid in these variable volume pipettes, there's a slightly different technique. You hold the pipette like this, and this knob here at the top is the one that you use to push the air out of the tip.
Okay.
Now, if you push down on the plunger, you'll find that after a very short distance it will stop. And if you press a little bit harder, you can actually push through that stop, and then you can push it down for a much larger distance.
So that's taking all the air out of the pipette and the tip.
Now, those two stopping positions are very important.
Mm-hmm.
The first stopping position is called the soft stop, and when you reach the soft stop, you pushed out exactly that volume of air.
Okay, so in this case we're looking to get 20
microliters.
Okay, yeah.
If you push further beyond that soft stop, then you get to the hard stop, and you've pushed out considerably more than 20 microliters at the hard stop. That extra push is when you are dispensing your liquid into a container.
Okay.
and it's designed so that every last bit of liquid leaves the tip.
Right, so when we're
all
drawing
of
things up, we're only going to the soft spot.
soft spot,
Okay,
yeah.
and the extra one is to blow out the
Is to
everything.
blow out
Yes, okay.
everything so that nothing is left in the
Okay,
tip.
well that makes sense now.
So to use these variable volume pipettes, you push down to the soft stop, 20 microlitres.
Yeah,
Put your tip in the liquid, just a couple of millimetres below the surface, and then gradually release.
okay, and that's important to not get any air bubbles inside there.
You can see that
etc.
there's no air bubbles in
Yeah,
there.
okay.
If you did get air bubbles in the fluid in the tip, then just push
Just
it all
blow it
out
out. Go
and
to the
start again.
to the hard one and blow it out.
Yeah.
Yes, okay.
Now, when you now dispense your liquid, let's put it back straight into the same beaker. You push this time all the way down to the hard stop. And you can now see that all the liquid has left the tip.
Yeah, okay. Right. Okay. And then that's another case of disposing of that tip because we
Yeah.
can't
Now,
be
after
using that
you've
again.
used that tip, okay, there is a trigger button here on the side of the pipette, and you use that to push the tip off.
Okay. Right, thank you.